Hey guys, welcome back. This time around we're going to take a look at a set of reactions known as the Krebs cycle. Now these are going to be 8 enzymatic reactions that are going to occur inside the mitochondrial matrix. They're going to be the first set of reactions that we've looked at in catabolism that are oxygen dependent. Now these set of reactions are known after the scientist who first identified some of the substrates and enzymes associated with it. His name was H.A. Krebs. He was German born and did most of his research in the 1930s in Britain. Now, the Krebs cycle is also known as the citric acid cycle because the initial end product will be a citric acid molecule, a 6-carbon citric acid molecule. The initial substrates will be 2 acetyl coenzyme A molecules that were generated during the transitional phase reactions. However, the first reaction is to cleave off that acetyl coenzyme A, which leads you to ask that question, why invest all the energy that we did in the breakage of the initial covalent bonds to create that bond between the acetyl and coenzyme A, if the first thing we're going to do is break the bond when we get it to the Krebs cycle? Well, the answer is twofold. First, the acetyl coenzyme A helps to carry the acetyl group to the Krebs cycle. Secondly, by breaking that bond, we generate the energy necessary to drive the Krebs cycle reactions. That bond is a very high energy bond, very similar to a phosphate bond. And by breaking it, we can release a lot of energy. It's an exergonic reaction that can be used to drive these reactions forward. Now, the Krebs cycle itself mimics what you would think of as an enzymatic furnace. We think of a furnace, we put wood in it, and we completely combust that wood, break it down to nothing but ashes. We garner the heat that was released by burning that fuel. Well, that's what's going to happen here. We're going to allow the acetyl group to go in, and we're going to completely combust it, break it down. So coming out of the cycle, there'll be no remnants of what's left of that acetyl group. In the process, we're going to try to take the energy that's released once we break the covalent bonds and the bond, the acetyl coenzyme A bond. We're going to try to take that energy and put it into usable energy in our body in the form of ATPs, or to store it away in potential energy in the form of NADHs and FADHs. Now let's take a moment and look at the big picture of the Krebs cycle. The big picture overview is that we bring in a 2-carbon acetyl coenzyme A molecule, and it combines to a 4-carbon oxaloacetic acid molecule to create a 6-carbon acetyl coenzyme A molecule that will be broken down during the cycle back to regenerating the oxaloacetic acid. Now, during each cycle, 1 ATP is going to be generated via substrate level phosphorylation. This is going to be our usable energy. 3 NADH molecules along with 1 FADH molecule will be generated, or potential energy, and it will be transported to the electron transport chain, remembering that those set of reactions occur in the inner lining of the inner membrane of the mitochondria, in those folds known as crista. Also note that 2 carbon dioxide molecules are going to be liberated. Now those 2 carbon dioxide molecules are going to be the carbons associated with that acetyl group that were brought in. Note that the initial 2 NADH molecules occur in conjunction with the cleavage of a carbon dioxide molecule. Now this is going to be a reaction that is governed or controlled by a protein complex. And that protein complex by definition is more than one enzyme working together in a cooperative way, in a quaternary structure, to allow a sequence or an assembly line type of approach to controlling the breakdown of a product. Now, the first step in that complex is going to be decarboxylation. Now, decarboxylation is the removal of the carbon molecule, the breakage of the carbon bond. That requires an enzyme known as a decarboxylase. Now, after we had the decarboxylation, subsequently we'll see that we'll take the energy that comes from that covalent bond and we will use it to fix or to reduce an NAD plus molecule to an NADH molecule. The enzyme that does that reduction is known as dehydrogenase. Now the FADH and the third NADH molecule is going to be generated using yet another dehydrogenase enzyme. Note that some of the energy released from the cleavage of the second covalent bond is coupled with the addition of an acetyl coenzyme A molecule to the succinic acid. Now, when we cleave the bond from that succinyl coenzyme A, we will use the energy from the breakage of that bond to help drive the remaining reactions. Also notice that this cycle represents only one acetyl coenzyme A. Remember though that when we started with this glucose molecule in glycolysis, we generated two pyruvic acid molecules. We took both those pyruvic acid molecules through the transitional phase and created two acetyl coenzyme A's. So for each glucose that we start with, we've actually got to take two acetyl coenzyme A's through this Krebs cycle. So really, if you're looking at the Krebs cycle, you got to remember that it's got to occur twice per glucose molecule. So everything that's produced in the Krebs cycle needs to be doubled. So let's do a summary then. Now we've learned though from glycolysis that we produce two ATPs, two NADHs, and we broke one covalent bond, if you remember, between the third and the fourth carbon of the glucose molecule, producing two pyruvic acid or pyruvates as our end products. During the transitional phase, we generated two NADH molecules, and we also generated two carbon dioxide molecules by the cleavage of two covalent bonds, producing a two-carbon acetyl coenzyme A molecule. And we take that acetyl coenzyme A molecules and run them through the Krebs cycle independently, but two cycles, we'll see that we generate two ATPs, we generate six NADHs, and two FADHs. Remember, we have to double everything that's produced in the Krebs cycle so that we take into account both the acetyl coenzyme A's. And we produce four carbon dioxide molecules. We cleave off two covalent bonds. The bond is going to be found between the two carbons of the acetyl groups. And by the end of the Krebs cycle, we have no remnants of the glucose molecule left. No covalent bonds are left. No carbon molecules are left. Everything's gone. Hydrogen's gone. The oxygens are gone. At the end of the Krebs cycle, then, we have generated a total of 4 ATPs through substrate level phosphorylation. We've generated 10 NADHs, 2 FADH2s, and 6 carbon dioxide molecules. We've broken all the covalent bonds. Where's all this potential energy being converted to usable energy? Well, it's going to be converted, as we'll see, in subsequent reactions in the electron transport chain and oxidative phosphorylation by taking advantage of the NADH and the FADH molecules that we generate in the previous 3 sets of reactions. You guys have a great day. I'll see you soon.